Good day to everyone joining us, and welcome to today's X-Talks webinar, Risk Management of Adventitious Agent Contamination in Biopharmaceutical Products. My name is Sari Abdo, and I'll be your host for today. Today's presentation will run for approximately 90 minutes, including a Q&A session towards the end. Today's webinar is designed to be interactive, so please feel free to send in your text questions and any comments throughout the presentation. You could do so by using the questions function that's located in the panel on the right-hand side of your screens. At this time, all participants are in a listen-only mode, and today's event will be recorded and made available for future download. The presentation slides will advance automatically, and if you need any further assistance, please send me a message anytime using the same questions panel. I would now like to thank Lancaster Laboratories, who have helped develop the content and presentation for today's meeting. With a proven track record of providing quality scientific solutions for 19 of the top 20 largest pharmaceutical and biopharmaceutical companies in the world, Lancaster Laboratories is a global leader in bio and pharmaceutical laboratory services, providing innovative and timely scientific solutions to streamline the drug development process. With a global capacity of over 300,000 square feet, Lancaster Labs has the abilities to meet your global regulatory requirements, and all of their facilities offer CGMP-compliant laboratory services and operate under the same strict quality control program and utilize the same LIMS system. For more information, please visit their website at www.lancasterlabs.com. I would now like to welcome our speakers for today's conference. Joining us today is Dr. Jerry-Ann Booz, who is the Director of Pharmaceutical Services at Lancaster Labs. In this position, Dr. Booz provides strategic, scientific, and regulatory direction for the company's biopharmaceutical testing business, which includes extensive capabilities in biochemistry, cell and molecular biology, and virology. She has expertise in the formulation of global regulatory product testing strategies and in the preparation of global regulatory filings at all stages of product development. Dr. Booz has earned a PhD from Georgetown University, and she has published numerous papers in the field and has held positions in academics, government, and industry. Also joining us today is the manager of the Viral Safety and Viral Clearance Services at Lancaster Labs, Dr. Catherine Bergman. Dr. Bergman has extensive experience in cell line characterization, lot release testing, and viral TSE clearance validation, and has numerous publications and presentations in the field. She has performed biomedical research in protein chemistry, cell biology, and virology at Georgetown University, Scripps Research Institute, and Rockefeller University. Dr. Bergman received a PhD from Brown University and an AB degree from Mount Holyoke College, and she has served on the PTA task force on preparation of virus spikes used for virus clearance studies. Our third speaker today is Dr. Yuling Li, who we're very happy was recently able to join our speaker panel. Dr. Yuling Li is currently a fellow and scientific director in process biochemistry biopharmaceutical development at MedImmune, where she is responsible for providing input on scientific and technical directions and strategies for the company process development activities. She is currently leading an integrated viral safety team across the organization to mitigate the risk associated with adventitious agents and also directing other cross-functional technical initiatives. Dr. Li received her PhD from Robert Wood Johnson Medical School. During the course of her career, she published over 30 peer-reviewed articles and is the primary inventor for 7 issued patents. She has spoken at many international biopharmaceutical conferences, and she was a member of the industry experts in task force groups working with the FDA, EMEA, and USP to address issues and guidelines associated with biopharmaceutical development. Dr. Li is the former president of the Chinese Biopharmaceutical Associations USA, a non-profit professional organization, and she also co-founded the Alliance of Chinese American Biotechnology and Biopharmaceutical Associations. Participants can access more information about our speakers and sponsors at the event's main page on www.xtalks.com. And now, without further ado, I'd like to hand the mic over to Dr. Bergman, who will begin with today's presentation. Dr. Bergman, you may begin when ready.
Good afternoon, everyone. Welcome to our seminar on risk management of adventitious contamination in biopharmaceutical products. Today we're going to discuss 5 main topics. First, we'll discuss assessment of raw materials for their individual risk for introducing adventitious virus contaminants into the process, manufacturing personnel and facility management, introduction of appropriate testing regimes, engineering appropriate adventitious agent inactivation and removal steps, and equipment and facility cleaning validation studies. First, though, a little introduction. An adventitious agent is an agent that is not intended to be present in the biological product. Examples of adventitious agents include bacteria, fungi, and molds, Mycoplasma and Spiroplasma, viruses, and TSE agents. Testing for these agents addresses one of the aspects of product characterization, and that is purity. Bacteria, fungi, and molds may be introduced through starting materials such as cell lines, animal origin raw materials, and plant-derived raw materials. May also be introduced by operators and using poor technique, and there are other possibilities as well. Bacteria are approximately 1 micron in diameter, and they can thrive in extreme environments, so they can be difficult to remove. Fungi and molds, in contrast, are approximately 100 microns in size. These agents are controlled usually by inactivation and by robust sterile filtration. And because of these controls, raw materials are usually not typically the source of any bioburden events that occur in the bioreactor. These agents, however, may be introduced by ineffective or incomplete facility or equipment cleaning, in particular any biofilms that can resist inactivation and cleaning procedures. Mycoplasma and Spiroplasma are considered bacteria, but unlike most bacteria, they lack a cell wall and they are much smaller. They also may be introduced through starting materials such as cell lines, animal origin raw materials, and plant-derived raw materials, and they may be introduced by operators. Mycoplasma are inactivated by heat, extremes of pH, and by gamma irradiation. And they also may be introduced by ineffective or incomplete facility and equipment cleaning. Biofilms are known to survive as high as 95 degrees C. Removal of these agents can be done through filtration through a 0.1 micron filter if that filtration is validated for matrix effects. However, it's not a sterilizing step because the agents are much smaller than the bacteria and they are not always quantitatively removed. E. coli Leishmaniae is a particular challenge. It's been found repeatedly in animal and plant tryptic soy broth, even after heating to 95 degrees and filtering. It's also been found in bovine serum, plant and animal peptones, and in water. Viruses may be introduced through the starting material as well, including cell lines, blood, other tissues, and they may be introduced through the use of contaminated raw materials. Bovine serum has been shown to have been contaminated with Cache Valley virus, reovirus, PI3, BVDV, and Vesivirus. Trypsin used for passaging of cells can be contaminated with porcine viruses, and other animal and non-animal, including plant raw materials, have been shown to be contaminated with viruses, including MMV as an example. These agents may also be introduced through operators. Examples have been found in manufacturing. And they may be introduced through contaminated facilities as well. Now, it's important to note that viruses are parasites, and they have to have a cell to replicate in, so they cannot replicate independently. They range in sensitivity to physical and chemical inactivation, and they range in size from a very small 17 nanometers up to approximately 200 nanometers or larger. TSE agents, that's short for transmissible spongiform encephalopathies, are typically introduced through the use of animal-derived raw materials. Ruminant origin materials such as serum And human raw materials and tissues such as blood and CNS tissue are known sources of this contamination. TSE agents are extremely resistant to physical inactivation, much more so than any of the other adventitious agents. And it is really best, if possible, to avoid use of these materials so that you don't introduce the risk of TSEs. If these materials need to be used, then the control over them is predominantly due to sourcing of the materials. And if necessary, clearance studies can be performed. All right, now let's turn to assessment of raw materials for their risk in introducing adventitious contaminants. The establishment of a risk assessment program should cover the following items: the likelihood of a contamination event, the ability to control the contaminant, what might be the consequences of the event, such as plant shutdown and cleaning, investigation, and possible cleaning studies. What is the impact on product safety, and what is the impact on product availability? A raw materials program should include a vendor quality audit. Attention should be paid, among other things, to the vendor facility pest control program and the vendor cleaning program. You should be reviewing the raw material manufacturing process to make sure that it is under proper control. And then each raw material should be classified as high, medium, or low risk for potential to introduce adventitious contaminants into the process. From this information, you should be developing a risk mitigation strategy for individual raw materials. This can include recommendations to the supplier for improvements, safety testing of the raw material, and/or addition of specific agent inactivation or removal steps to the process. And then also you should develop a risk mitigation strategy for strategic combinations of raw materials such as media components. All right, our next topic is manufacturing personnel and facility management. This will include things such as storage practices for raw materials, in-process materials, and finished products to make sure that there is no risk of reintroducing anything into these steps. There should be a facility pest control program and a facility cleaning program. And with regard to the manufacturing process, you should pay careful attention to the personnel and facilities, ensuring that the process is under a risk continuum flowing from high to low risk with physical and personnel barriers at strategic locations. This will ensure that after the product has been cleaned up, there's no opportunity for an adventitious contamination to occur again. You'll need to keep in mind improvements to the mitigation strategy during manufacture as new information comes online about risks and processes may change. And you will want to engineer safety into the process at the time of use via testing and adventitious agent inactivation and removal procedures. So with using all of these features, you want to develop an in-process, real-time manufacturing testing plan so that you can tell as the process is going on if you're having any contamination occur. At this point, Jerry Ann will take over on discussing the next subject.
Good afternoon, everyone. The part that I'll be covering today deals with the introduction of appropriate testing regimes for a battery of adventitious agents at strategic locations within the manufacturing process. Within this schematic, I'll be emphasizing the testing that should be performed on master cell banks, working cell banks, the unprocessed bulk material that is derived from manufacturing, as well as the associated end-of-production cells, and we'll also talk about the purified bulk product and the final vialed product as well. So let's begin with the cell bank testing plans. A few definitions and a few generalizations about the testing that should be performed. The master cell bank is the starting material for the whole of the production process. As such, you want to spend a great deal of time fully characterizing this bank for microbial and viral contaminants. This is one-time testing that will serve you for the lifetime of the master cell bank. The working cell bank is just a few number of passages beyond the master, and as a result, it is possible to have a reduced package of testing. Cells at the limit of in vitro cell age, or the end of production cells, and I will take a step back and say that although there are subtle differences between these two phrases, the industry has started to use them synonymously. These are the cells that are left at the end of the bioreactor run, or which are taken several passages beyond. They are considered to be worst case for amplification of contaminants as they have experienced the stresses of the bioreactor. So again, we urge you to do full characterization of these cells, and it needs to be done only one time unless the bioreactor culture conditions change. The master cell bank and end of production cell bank testing should include sterility, Mycoplasma testing, and a number of different assays for viruses that will include broad specificity assays, species-specific assays, and a battery of assays for retroviruses. So a little bit about each of the assays that we would recommend. The sterility assay is a pharmacopoeial method that involves direct inoculation of the cells, typically into two media under aerobic and anaerobic incubations for a period of 14 days. During the course of the assay and at the end of the assay, the medias are observed for turbidity. For cell banks, the requirement is that you test 1% of the total bank or a minimum of 2 vials. And we also urge you to perform bacteriostasis and fungistasis testing to assess the sample matrix for inhibition. Mycoplasma are common cell culture contaminants. Common sources for the introduction of mycoplasma into cell culture systems include the cell culture media and additives, the cells themselves, and laboratory personnel through a breakdown in GMP. Mycoplasma contamination of cell lines can lead to adverse cytopathological events that might include altered cellular metabolism, disruption of nucleic acid synthesis, changes in gene expression, and changes in cell membrane antigenicity. The bottom line is that mycoplasma are associated with disease, and mycoplasma infections may impact the yield and quality of biological products. For this reason, mycoplasma testing is a regulatory requirement for biopharmaceuticals as it supports product safety. Mycoplasma assays for biotech products are typically performed using an assay that's harmonized per the conditions laid out in points to consider documents, the European Pharmacopoeia, and the US Pharmacopoeia. The testing procedures for such a harmonized assay are 28 days in duration, and they have 3 components. Detection on indicator cells, this is the short portion of the assay. Direct cultivation of the sample or application of the sample onto agar plates. This lasts for 14 days. And then probably the most sensitive part of the assay is incubation of the sample in broth to allow amplification of the low-level contaminant, followed by agar subculture. This is the portion of the assay that is rate limiting and in fact does last the full 28 days. As with bacteriostasis, fungistasis for the sterility assay, we urge you to perform mycoplasma stasis to assess the sample matrix for inhibition. A hot topic in mycoplasma testing today is the application of rapid methods to replace this long 28-day assay. These methods are well accepted in Europe as lot release testing methods. That is not the case, however, in the US. At this point in time, work is underway to make these rapid methods suitable for lot release testing in the US. Currently, however, our clients based in the US do a great deal of rapid testing as part of an in-process check to determine if a Mycoplasma contamination event has occurred. So when I start to think about viral risk assessment of a particular cell line, I typically begin by asking our clients a lot of questions. I want to learn as much as I can about the origin and history of that cell line. Typical questions that we'll ask will include the following. What is the origin and history of the cell line? Has it been exposed to serum or trypsin? And I mean not only when the client is working with that particular cell line, but over the period of the total history of the cell line. Has the cell line been exposed to other cell species or to substances derived from other cell species? And what non-animal additives has the cell line been exposed to? A lot of people think that if you substitute animal additives for plant-derived additives that you are home free, but that is truly not the case. We need to also pay a great deal of attention to what viruses the cell line might be susceptible to. With all of this information, we can then identify the risks and develop the testing and response plan to a contamination event. Now, if the master cell bank has been exposed to serum, trypsin, or any other animal-derived additive, they should be certified to be virus-free using the 9 CFR bovine and porcine assays. If the serum or trypsin are continually used to culture the cells, a 9 CFR screening of each lot of serum and trypsin used is part of a good GMP production program. And in a couple of slides, I'll go into detail about how to perform the 9 CFR assays and what viruses these assays will test for. The bullet points at the bottom of the slide are simply to reiterate some of the more common contamination events that have been introduced through the use of serum, trypsin, and other media components. Now, there's no one viral assay that we can use to look for every virus that's out there. So we really have to attack looking for adventitious viruses through a battery of assays. And these include broad specificity assays, both in vitro and in vivo. And I'll say a word about electron microscopy here. I can't emphasize enough that an electron micrograph can be of enormous value in the identification of an unknown contaminant. The micrograph will reveal the physical characteristics of the virus as well as its location within the cell. And these are great clues as to what we're dealing with. Another set of assays that we'll perform include the 9 CFR assays for bovine and porcine viruses, and then there are other assays for rodent and human viruses as well, which will detect viral contaminants associated with specific species. And finally, we come to the retroviruses, which I know many of you out there are familiar with, especially if you're working with rodent cells such as CHO or mouse. There are several different types of assays that we can use to look for the presence of retroviruses. These can be morphological, it can be by infectivity, or it can be through biochemical analysis. So beginning with the in vitro viral screening assays, these are based on the principle that viruses will replicate in a suitable host cell. Presence of the virus is detected by the appearance of cytopathic effects and/or hemagglutination or hemadsorption of specific types of erythrocytes. The indicator cell lines that we use in the assay are selected based on the theoretical susceptibility to viruses of concern. So here's where we've done a risk assessment, and we are predicting what viruses could possibly be introduced. That helps us to select the 3 cell lines. In almost every instance, and I quite frankly have never run one of these assays without the first 2 cell lines, we'll use a human diploid cell line, MRC5, and a primate cell line, viro cells, to detect viruses that would be infectious for human cells. The third indicator cell line should be a monolayer culture of the same or similar cell type as the cells you are using in manufacture. This cell line will detect viruses that are infectious for those cells. So for example, if you're using a CHO cell line in manufacture, we've got CHO K1 cells available for testing in this assay. And also, many of our clients will elect to add additional cell lines to this assay. If you're extremely worried about murine minute virus, for example, you may decide to add A9 or 324K cells to ensure that you will detect this virus. To perform the in vitro assay, the indicator cell lines are exposed to the test article, and the cells are incubated for 2 to 4 weeks and observed microscopically for the appearance of CPE or cytopathic effects. The test cultures are compared to negative and positive control cultures, and at the termination of the assay, either 14 or 28 days, and whenever you're testing a cell bank, I urge you to do the 28-day assay to detect that low-level contaminant. Hemagglutination and hemasorption are performed. We recommend that you do matrix interference testing and that you do this by analyzing spiked model viruses at various levels, by spiking model viruses at various levels into the test article. The in vivo assay for adventitious viruses is designed to complement the in vitro screen I just described. The principle here is that viruses that do not cause cytopathic or other noticeable effects in a cell culture system might be detected in an animal system. The typical animal systems are listed below, and during the time course of the study, the animals are observed daily for clinical signs, typically just evidence of good health and survival, again, every working day of the specified test period. The bovine and porcine 9-CFR assays fall under that category of assays designed to detect species-specific viruses. The basis of the assays are to detect a wide variety of bovine or porcine viruses based upon development of cytopathology, hemasorption of red blood cells, and specific immunofluorescence staining. In short, the sample is inoculated onto the indicator cells, which are then monitored microscopically for cytopathic effect for 21 days with 2 subcultures. And at the end of the assay, the cells are tested for hemasorption, and the cells are then fixed and stained with antibovine or antiporcine virus fluorescein-labeled antibodies. I show you a list of all of the viruses that will be detected in the bovine 9-CFR assay, and the next slide shows the same for the porcine viruses. Additional species-specific tests include the antibody production test for the detection of rodent viruses. These assays are very sensitive methods for detection of adventitious rodent viruses and involve the use of viral antibody-free animals. These animals are injected with the test article, and then serologic analyses are performed at the end of the incubation period to determine if antibodies were produced against specific viruses. The test may be performed in 3 different species, mouse, rat, and hamster, and the species selected will depend upon the species of origin of the cell line that you wish to test. A summary of the procedure involves inoculation of the cell lysate into the animal via a variety of routes. The different routes will emphasize or be preferential for different types of viruses that you are looking for. The animal is then bled after 28 days and tested for the presence of virus-specific antibodies by ELISA and or immunofluorescence staining. Now, it's really quite important to be aware that not all of the viruses that you might be concerned about will be detected in the assays that I've described so far. And so we urge you, as you're doing your risk assessment, to make sure that if the viruses you're concerned about are not going to be detected in these assays, that you consider the addition of virus-specific qPCR assays to your testing panel. And some of these examples would include vestivirus, Cache Valley virus, porcine circoviruses, and you might want to also include PCR panels that are specific for a variety of different species depending on your the species of origin of your cell line. So the final category of viral tests includes assays for retroviruses. Although these viruses are not considered adventitious agents, they might be endogenous to your manufacturing cell line. And anyone working with CHO cells or with mouse cells is very well aware of the concerns around these types of viruses. Specifically, things that might happen if retroviruses are present include insertion of the virus into the recipient cell genome, and this can result in transformation and tumorigenicity, as well as inactivation or overexpression of the gene, and ultimately it can result in cell death. Retroviruses are associated with serious immunosuppressive and degeneration conditions, and therefore contamination of your product with retroviruses is of major concern to the industry. So it's necessary to demonstrate the absence of infectious retroviruses and to demonstrate that the downstream purification processes will adequately remove these viruses from the production stream. In considering which retroviral assays are appropriate for your different situations, keep in mind that many retroviruses have the capacity to remain latent within cells. And importantly, not all cell lines are susceptible to a given retrovirus. In fact, many retroviruses will also infect and replicate in the absence of obvious cytopathology. So to address these issues, a series of specific assays and co-cultivation procedures based upon cytopathology are available to detect a wide variety of infectious retroviruses. I've listed some of the commonly used retroviral infectivity assays. I won't go through them in detail. Suffice it to say that the three most common assays for murine leukemia viruses are listed above. These viruses rarely produce obvious cytopathology in vivo, with the exception of within these test systems. I've also listed some commonly used co-cultivation systems which utilize susceptible target cells of murine, mink, and human origin. And again, I would emphasize that these lists are partial. In addition to the infectivity assays, there are a number of alternative assays that can be used for retroviral detection. The most common is to test for an enzyme that is found only with retroviruses, and that is reverse transcriptase. It's also possible to assay for the presence of retroviral nucleic acids and retroviral proteins. And I think many of you are already common with the use of electron microscopy to determine the presence, intracellular location, and morphology of any retroviruses or retroviral-like particles that may be present. So coming back to a slide we've seen before, the rationale for cell bank testing, we've covered the different assays that we'll use in this testing, and we've gone through the extensive assays that we'll use for the master cell bank, as well as the end of production or cells at the limit of in vitro cell age. I'd also mentioned previously that because the working cell bank is just a small number of passages removed from the master, that we have a reduced package of testing, and that is shown here. This slide, again, lists all of the assays that we'd use for the master or end of production cells. The assays that are highlighted in orange, a subset of the assays, are the only assays that you need to use to test the working cell bank. Moving on to testing of the unprocessed bulk harvest, what is required here is microbial testing of all of the bulks for bioburden or sterility. You'll also need to test all of the bulks for Mycoplasma, as well as all of the bulks should be tested for the in vitro adventitious viral screening assay. In addition, you should consider, or you may be required, to test your cells, your unprocessed bulk rather, for infectious retrovirus. You might want or be required to do in vivo testing, and you should certainly, as we mentioned before, consider PCR assays for viruses of specific concern. You will also want to do test three consecutive bulks for retroparticle counts by TEM analysis, and this is for the purposes of setting your retroviral viral clearance goals. I'd also like to discuss in-process testing at the bioreactor stage of the manufacturing process. It's an excellent location at which to consider the implementation of in-process tests that will provide early indications of adventitious contamination events. The bioreactor is a key location for this testing, as bioreactor conditions may promote the growth of a low-level contaminant not previously detected during the testing of your raw materials and cell lines. In-process testing methods would include monitoring cell viability and growth, monitoring critical bioreactor parameters such as oxygen demand, and the use of PCR and other rapid testing methods for specific adventitious viral agents. And I went back to a lot of information I've gathered for contamination events that I've been involved in, and can give you a couple of examples where cell death in the bioreactor really tipped us off as to some contamination events with Cache Valley virus and with human adenovirus. Poor cellular growth tipped us off as to contamination events with murine minute virus and vesivirus. And in contrast, with reovirus type 2 and porcine circovirus type 1 contamination events, there were no observed effects on cellular growth, product quality, or yield. So in those situations, I would really urge you, if you're worried about those viruses, to have a rapid testing method specific for them. We do very little testing on the purified bulk and final vialed product. For adventitious contaminants, really all we do here are looks for microbial contaminants through a sterility test and an endotoxin test. And this is because most of the testing for adventitious contaminants is performed prior to purification so that we optimize the detection of those contaminants. So this concludes my portion of the presentation, and what we've really done is we've talked about the control and testing of the source materials. Kate talked about the raw materials. I talked about the cell substrates. We've talked about the testing of product and intermediate product at the appropriate stages of production. And these are all very important steps to take towards mitigating the risk of adventitious contamination. However, it's not enough. The steps that we've described so far are not without their limitations. First and foremost, testing will not detect all viruses. Quite frankly, we don't even know all of the viruses that we need to be worried about. New viruses are being discovered every day. Some of the direct testing methodologies are also so specific towards a particular strain or variant of a known virus that we might miss a new variant or strain. And finally, one of the biggest concerns is that the direct testing methods might not be sensitive enough to detect virus that's present at very, very low levels. And we also, when testing large lots of material, need to be concerned about the sampling size issues and the statistics of sampling. So how do we ensure that there is no risk of viral contamination? With that, I'm going to turn it back to Kate, and she's going to describe two additional approaches that should be taken.
Okay, so we'll be discussing engineering appropriate inactivation and removal steps next. There are a variety of steps that you will be using during purification of your product, and all of these have potential capability for removing adventitious agents as well. So there are a variety of removal steps such as virus filtration, column membrane chromatography, and precipitation steps. And then there's a variety of things that you can do that will inactivate virus in particular, such as heat, irradiation, pH, and so on. Sorry. So it's important to incorporate robust virus removal and inactivation steps during the design of the manufacturing process at the earliest stages of process development. You don't want to develop a process, purification process in a vacuum, so to speak, and then find out months or perhaps even years later that you don't have sufficient capability for virus removal and inactivation. And it is important, having designed your process with this in mind, that you still need to test the steps to make sure that they're effective in removing or inactivating viruses. And one of the terms that's used for this is viral clearance studies. There are a variety of other names that have been used, viral validation, process validation for viral removal. But for the rest of this talk, I'll just use the term viral clearance studies. Viral clearance studies are a complementary strategy to the testing that Jerry Ann's been describing. It's kind of a what-if experiment. If the viruses are present in the starting material, can they be removed during purification? So the way this is done is that the purification process itself is evaluated for its ability to remove and or inactivate specific viruses. Because each purification process is unique, each virus clearance study is a custom study. It will be customized to the particular process that you've developed. And the way this is done is that individual steps that are selected are assessed separately for viral inactivation or removal. The calculation of the virus clearance achieved is based on the log of the virus that it was in the material to begin with, divided by the log of the, or divided by the virus that was in the output. So that you're looking at a log scale here that implies, among other things, that you're not promising that all the virus is removed. You're just removing certain factors of the virus. The objectives of a viral clearance study are first to provide evidence that the manufacturing process has the capability to effectively remove and or inactivate viruses that are known to contaminate the starting material or that have a real possibility of contaminating the starting material. In addition, we'll want to look at the production process for its ability to remove or inactivate any virus in the event that a novel or unpredictable contamination occurs. And the results of both of these approaches provide assurance that any virus that enters at any point during the process would be removed during the purification process. It's important to select the correct viruses for use in the clearance study. Viruses that represent viruses that could contaminate the starting material should always be included. These are termed relevant viruses. Typically, the virus panel should also include viruses that provide a wide range of physical and chemical characteristics so that you can evaluate the performance of the process at clearing viruses that are very different from each other. The mechanism of action to inactivate or remove the virus should be considered, and the most resistant viruses for those steps should be included. The resulting data from these studies will provide confidence that the process is able to clear unknown or unpredictable adventitious contaminants. There are a variety of assays for quantitation of virus. The standard assays that are the most commonly used are infectivity assays, and there are several kinds of those. There also are quantitative PCR assays that can be used for this purpose. However, you do have to keep in mind that the PCR assays are detecting the viral nucleic acids, and you have no way of knowing when assaying that whether the virus is infectious or not. You only know that there is presence of the viral nucleic acid. Whatever assays are selected should be validated to confirm that they're sensitive, reproducible, and that they're performed with sufficient replicates to ensure statistical accuracy. Every assay should also include appropriate negative and positive controls to demonstrate that the method is meeting its predefined acceptance criteria. And all of the materials used in the assays, including indicator cell stocks, virus stocks, and assay reagents, should be well controlled to ensure the reproducibility of your assay. In the purification process, there are obviously quite a number of steps that are used, and you want to look at the overall production process and evaluate which steps should be used. It's not required to assay all of the steps. You want to look at the steps that are most likely to provide clearance, that are readily and reproducibly scaled down, and very important also that they have independent viral clearance mechanisms, that they not be clearing virus by exactly the same method. It's in your best interest to emphasize the testing of the robust steps. Those are the ones that clear a wide range of viruses without being influenced by process variables. And you want to define and understand the mechanism of clearance for each step, whether it's by inactivation, by removal, or a combination of both. Now, returning to my slide from earlier on a variety of steps that are used in purification processes, let's go into a little bit more detail on some of the steps, starting with the removal steps. Viral filtration is now one of the most common steps used in bioprocessing. It's a very rare purification process that does not include a viral filtration step. The only purpose for viral filtration is to remove virus. It does not purify the material in any other way. It is a single-use technology, which has the benefit that there's no need for reuse and cleaning studies. It's highly robust when the process parameters are well understood. The mechanism is based primarily on size, and clearance is therefore dependent on the filter used and the size of the spiking virus. Chromatography, whether done in a column format or a membrane format, is not generally considered to be robust with regard to viral clearance. The reason for that is that there are a number of parameters that can vary that can have a significant effect on clearance, such as pH, salt concentration, flow rate, protein content. The clearance that's achieved on these steps does support that for the robust steps, and it is heavily relied upon by the manufacturers to increase the clearance number. But it is important to remember that it is condition dependent, and if the conditions change, the clearance could well change. The surface chemistry of the virus is a critical factor in the clearance. There are a variety of chromatography steps that can be used, such as affinity, anion exchange, hydrophobic interaction, size exclusion chromatography. We haven't mentioned before, but viral clearance study is usually carried out in several stages, and the early stages are a more limited scope of study than the later stages. And in the late stages, there are several things that you need to make sure you add to your program, such as mass balance, evaluation for virus, column reuse, and cleaning studies also should be included in your consideration. Precipitation methods are also can be useful as virus removal steps. However, they do have some significant disadvantages. They have significant scale effects, and that makes them very difficult to scale down. They're not generally robust because the critical variables are not well understood. And it can be difficult to determine what the worst case scenario is. And there is definitely a concern regarding the entrapment of virus in the precipitate that is more or less artifactual, and which can result in an underestimate of the virus remaining from the precipitation. The virus surface chemistry is also quite important in this process. So a precipitation method provides supportive clearance data, but it's not something that you would prefer to rely on for the majority of your clearance. Now we'll move on and talk about inactivation steps. Oops, one slide too fast. All right, there's a variety of inactivation steps that can be used: heat, radiation, pH, solvent, detergent, and then there's two newer methods that I'll discuss in a minute. Viruses can be separated into two main categories. The first is viruses that have an envelope, and the second is viruses that are not enveloped. The virus envelope is attached to the virus as it buds through a cell envelope, and it's very similar actually to the envelope of a cell. So it has lipids and the kinds of surface proteins that a cell membrane would have. So envelope viruses are generally considered to be low to medium resistant to physical chemical inactivation such as heat and irradiation, whereas non-envelope viruses have a high resistance to such things. With regard to solvent detergent treatment, envelope viruses are sensitive and non-envelope viruses are resistant. For pH treatment, the envelope viruses are sensitive both to high pH and low pH, and with non-envelope viruses, they are also sensitive to high pH and low pH, but you have to go to more extreme pH conditions in order to achieve inactivation. I know of some viruses that are stable at pH 11, so you can see that you have to go pretty far to inactivate a non-envelope virus. And examples of viruses of these categories are listed at the bottom. Now, most of these commonly used inactivation procedures preferentially inactivate lipid envelope viruses, and they can be highly robust for those viruses under defined conditions. But it can be very difficult to find an inactivation procedure that's effective on non-envelope viruses, but is also compatible with the activity of the product. So in more recent years, there are a couple of new inactivation procedures that are demonstrating efficacy with regard to non-envelope viruses. And while they aren't yet in the commonest of use, they are very promising for processes that yet to be developed. The first of these is HTST for short. That's high temperature short time. And in an HTST procedure, the solutions are exposed to temperatures greater than 90 degrees C for very short periods of time, milliseconds to seconds. It's high enough temperature to inactivate the viruses and a short enough time that it does not alter the properties of the material that you're treating. As you can see from the list, there's some very significant clearance factors that have been measured for a variety of different non-envelope viruses. This procedure works very well in a flow mode, a flow-through mode. In fact, that's about the only way you can manage it. And so it's become a very common way to pre-treat liquid raw materials prior to addition to the bioreactor, because it can just be put in the flow path and not interrupt the pace of the procedure. The other procedure that's becoming more common these days is UV inactivation. There were many attempts back 10, 15 years ago to use UV irradiation as an inactivation method, but it wasn't very productive. In the first place, there was incomplete virus inactivation of both enveloped and non-enveloped virus. People who tried it found excessive damage to their product, and anything that was done to protect the product also had the property of protecting the virus. And it was not very easy to scale, either down or up. More recently, several manufacturers have come up with improvements in the UV technology. The first improvement is to use monochromatic UVC light at 254 nanometers. This targets primarily nucleic acids and spares the proteins, which is why it's not as damaging to the product itself. The second improvement was an efficient mixing procedure so that all of the product stream receives a uniform UVC dose. As it happens, UVC doesn't penetrate very deeply, so that if the product stream is very thick, the molecules that are close, the particles that are closest to the UV light will receive a higher dose than whatever's on the farther away from it. One of the most useful things about UVC is that virus susceptibility to it is correlated to genome size, with the smaller genomes being more susceptible. And many of the most difficult viruses to inactivate are the smaller ones with the smaller genome. So this procedure targets specifically the viruses that are the hardest to inactivate. And it's quite possible to achieve greater than 4 logs of clearance with non-envelope virus. Now, I spent quite a bit of time talking about viral clearance studies. There are several other clearance study types that are used less commonly, but there are occasions when they are needed. As I mentioned a while ago, if you're working with a material that has the potential for TSE contamination, then a TSE clearance study may be necessary. It's not something you want to do if you don't have to, because it's a very long study. It's performed in animals, and it is both long and complicated and therefore expensive. There are special circumstances under which Mycoplasma clearance or bacterial, fungi, mold clearance studies may need to be performed as well. All right, turning now to our last topic, cleaning validation studies. It's important to ensure the freedom of your starting equipment and facility to the freedom from adventitious agents, and particularly in a multi-product facility where you're doing frequent changeovers, you need to be sure every time before you start a new project that you have cleaned all of your equipment and your facility adequately. There are a variety of ways these studies are conducted. The easiest thing to do is a solution phase study where you just test whatever agent it is you desire in the solution of your cleaning reagent. However, this is kind of the best case scenario and the one where you're the likeliest to get good inactivation, so it's not really very representative of the manufacturing situation, and it's not considered acceptable for a cleaning study. The more acceptable studies are all done on the surfaces of the equipment that you're using or something that represents it. And you may be looking for both inactivation and removal for this kind of study. Certain kinds of studies can be done using clean-in-place or automated cleaning procedures. But if you need to do an actual spiking study where you're spiking the agent, you obviously are not going to be doing that in your manufacturing plant. And so instead, you'll need to use coupons that have the same composition as your stainless steel bioreactor or your countertop or floors or walls or something, but it's not actually in your plant. You can operate it under appropriate safety conditions in another area. You need to pay attention to a worst case study design, which includes the agent you're testing being dried on the surfaces. The cleaning reagents you need to look at for the type, the concentration, and the preparation methods. And you need to look at contact time and temperature. The most common agents used for these kinds of cleaning studies are bacteria, fungi, and molds. However, there is an increasing use in these studies for Mycoplasma and viruses, and also for TSE agents. Now, consideration for a coupon study design, the coupon should represent the surface being cleaned. So if you're worried about your stainless steel bioreactor, you need to be working with stainless steel, ideally the same kind of stainless steel. The manufacturers of your equipment usually can provide you with a source for obtaining these coupons. The spiking agent should be resuspended in the matrix that's representative of your product intermediate, because the presence of the product intermediate can affect the ability of the cleaning reagent to inactivate or remove your material, the agent, sorry. The coupon should be spiked with the agent and taken through a process that mimics as best you can the actual cleaning process. And the cleaning reagent should be tested at or slightly beyond the shelf life, at or below the cleaning temperature, and at or below the lowest concentration used, all with the intent of monitoring the worst case scenario. You should consider evaluating kinetic samples, that is doing a time course through the process. You should consider whether or not you need to look at mechanical removal or just inactivation in situ, so to speak. And it is always necessary to perform interference testing in advance of the cleaning validation study. Many of these agents are very deleterious to the standard assays that are used for these agents, so you have to come up with concentrations that you can use acceptably, and you may need to neutralize the cleaning reagents as well. Okay, so this completes the part of the presentation from Lancaster Laboratories. We've tried to show you a package of procedures and techniques that will assist you in developing a risk mitigation strategy. We've tried to show you where to identify the risks, how to eliminate them, and kinds of tests that you can use on the contaminant. You should keep this risk management approach in consideration from the beginning of the product lifetime and continue through to the end. Any changes that you make in your process, you'll need to evaluate for changes in possible risks and or mitigation of adventitious contamination. So our goal here has been to provide you with the information you need to develop a robust prevention and plan and a response plan for adventitious contaminants. And we've provided a general overview of all the considerations in this topic. At this point, we're going to turn the session over to Dr. Yuling Li from MedImmune, who's going to present to you a case study of how this works in an actual manufacturing situation.
Thanks, Kate. Thanks to both Kate and Jerry Ann for very comprehensive overview of a testing point of view and process understanding point of view, and use those tools effectively to design into the process. So what I'm going to do is to share some thoughts from a biopharmaceutical production manufacturer point of view in looking at risk mitigation on adventitious virus contamination. And I will share some thoughts from first, we capture, next slide please, the risk associated with adventitious viruses. And look into an integrated viral safety strategy, taking the capability from analytical testing side of it, and also look into ways to build in additional viral barrier in the raw material program, and work in the viral removal and inactivation capability, which has been described very comprehensively by Kate, and overall look into the control and prevention infrastructure. And then I will also share some thoughts about opportunities and challenges associated with developing such a program. Next, please. So why we need to really think about a risk mitigation on adventitious virus contamination. As a biopharmaceutical manufacturer, we have the responsibility to ensure product quality and to protect patient safety. It's very important that we have a very good assurance that our products are free from adventitious virus contamination, and in general, adventitious agent contamination. And as in one of the most regulated industries, we had regulatory compliance we need to be adhered to. And also, viral safety is related to protecting our personnel, protecting our facility, and also protecting our business, so that we can really also ensure project progression. Next, please. Next. So, if we can move back to one slide, to slide 4. Marianne mentioned earlier, virus is a parasite, and the risk in a cell culture production process really is associated with our commonly used cell line, such as the CHO cell line, the N0 cell line, the Viral cell line. It's susceptible to viral infection. And once they got attached by virus, and virus effectively utilize the cellular machine to produce a lot of them, and that can lead to extremely high titer virus preparation. And with extensive control and preventive safety measures, many of them have been shared in the previous presentation, the recombinant DNA protein product derived from mammalian cell lines has a very long history of viral safety for patient use. However, there has been rare events of viral contamination that cause a severe impact to the company on product supply, production facility downtime, and lead to significant business loss. So I would like to share a quote from Tony Lubinicki of Johnson & Johnson. The virus is ubiquitous in nature, and no single precaution is foolproof. Therefore, we need to look into our strategy thoroughly and really assess the risk. And here is a table that was adapted from Mike Wiebe and other conference presenters from the PDA conference in December 2010. And that listed some published industrial viral contamination events. And if we look at the table here, starting from late 80 and all the way to 2010, there have been lots of events that have been reported, and some of them has huge impact on patient supplies and also really impact the business. And look at the kinds of virus that we're dealing with. You can see mouse minute virus, MMV, has occurred several times in this table, and the Wesley virus, highlighted in orange, that have caused Genzyme's product and production facility impact in quite a significant way. So with that, what we need to do as a biopharmaceutical manufacturer, we want to take a look of the risk associated with our current practices and develop the best strategy to put us into a better mitigated state. Next, please. And in an ideal state, we would like to look into 5 different areas to mitigate the risk associated with adventitious virus contamination. And as mentioned earlier in the previous presentation, the impact of viral safety started very at the beginning when we first make the cell stock, generate a reagent, create a DNA for the cell line transfection. Because if at that point, if we had used any agent that can carry potential risk to the cell line, that risk associated with the entire product life cycle of the program. Therefore, it's very important, and particularly for those who are in the research area at the time when we're generating the cell line, you actually really don't see the light associated with a particular product. However, from that point on, if you generate a cell line, the history will associate with the product. And therefore, it's very important for those of us who are at the front end of the program, carefully consider what reagent we use, and also carefully document what reagent we have used and the grade and the origin of the material and the safety assessment associated with those materials. And from a cell line on, if you look at the middle side of the graph, that's where we start to generate a cell bank and then get into the use of the cell culture process in the bioreactor and go through the purification and eventually lead to the production of the final drug product. And within that process, there's several areas that we need to think about. One is the virus testing that has been discussed extensively in the previous discussion, and testing the cell bank, the master cell bank, the working cell bank. and also test the in-process sample when the unprocessed bulk was harvested from the bioreactor. There's a testing that's required. And at the same time, more and more company is looking into a pre-harvest testing that with a specific high-risk agent through a specific qPCR testing or similar kinds of testing to assure that you had a pretty good safety assessment before the bioreactor is opened up for further processing, and also material gets spread to a broader side of the facility. And another area it's important to think about is to develop the infrastructure to implement good procedure, which include personal management, get necessary training, and have appropriate gowning procedure, and look into the facility design, the process, the flow of people and the flow of material, and the air filter, and all kinds of associated mechanisms to make sure that things are in the good place and really ensure a very good viral risk mitigation philosophy. And similar to what Kate described, is the plant cleaning. One would like to know, is your current cleaning procedure effective in a case of cleaning up viral-related contamination? And make sure that the procedure is appropriate in routine use, and also having procedure in place to for a rare case that when decontamination is necessary. And as mentioned earlier, whatever we put into the bioreactor, because the cells can be effectively used by the virus as a cellular machinery, anything we introduce into the bioreactor needs to be controlled. And in terms of control, we can consider two areas. One is really knowing what you're getting from your source. So it has a very important to have a good sourcing control, and audit your raw material supplier. Make sure they have their good practices in their warehousing, their transportation, and their change control and change over procedure in between products. And well, it's appropriate to have a process that can avoid animal material. And if it's important to use animal-derived material for the program, then use treated raw material as appropriate. And also, more and more companies are adapting into a media barrier protection, where raw material can be treated by high temperature short time treatment or by irradiation. And some of the more sensitive material that cannot be used by either of those methodologies, a nanofiltration methodology can be adapted to, so that you can really cut down the potential introduction of adventitious agents before the material flow into the bioreactor and get into contact with the cell. And another important barrier to ensure product safety is the viral clearance strategy, which Kate has done a very eloquent description about removal, inactivation, and I would not need to repeat it here. So overall, this is a ideal control strategy that can be put in place to have a very thorough coverage and control on the viral risk. Next, please. So our goal is to effectively use the analytical barriers, the raw material barriers, and the control and prevention infrastructure, coupled with robust viral clearance capability in the purification process, to have an effective risk mitigation strategy. Next, please. And having those combination of barrier will help to protect our production infrastructure. And at MedImmune, we executed those risk mitigation strategies through effective team structure. We had a company-wide steering committee and oversee different sub-teams to carry on the execution of those strategies. We had infrastructure sub-team, we had raw material barrier sub-team, viral detection and identification sub-team, and we also had a team to look into viral cleaning, effectiveness, and viral clearance. And we also had a team to keep an eye on the regulatory requirement, the industry experience, and keep us up to date in terms of the viral safety considerations. Next, please. And so I would just like to walk you through with some specific example in how we develop those viral barrier. And the one is the raw material barrier. As I mentioned it is important to have a comprehensive assessment of risk associated with the raw material sourcing and its use. And the sourcing evaluation and control, including avoiding animal-derived material when it's possible and use treated or inactivated components when it's appropriate. And have appropriate screening and testing procedure in place. And understand your vendor and conduct vendor inspection and qualification. And develop the media barrier to address the key risk of very low level virus could come from raw material that cannot be tested. And high temperature short time treatment for heat stable large volume solution is a very good strategy. And also we need to identify appropriate viral embedment technology for heat label components. Next, please. And so we conduct a proof of concept test, several study on proof of concept test. And we use a two-platform cell culture production process with our MedImmune proprietary media and also proprietary feeds, and test the conditions with untreated media and a condition that's reported in common literature treatment condition, and then increase the exposure 50% more, and look into the performance of a model cell line. Next, please. And what we see here is in an overgrowth cell culture performance, we can see the untreated, one-time treated, and 1.5 time treated media deliver very similar performance for a batch culture overgrowth condition. Next, please. We then evaluate the platform process, and when we see it, and in the top left graph, you can see a cell line that is in fed batch process X. We can see the viable cell concentrations versus time deliver very similar performance among the group, and the process titer, product titer versus cell culture time also deliver very similar performance. And in another platform process, give us a very similar indication in terms of cell concentration versus time. And then on the fed batch process, we treated the basal media and the fed media in a various combination in DOE study. And we can see that the product titer deliver very similar among all experiments. And it gives us the comfort that the condition that we use to treat the media and the feed media were very capable of delivering the cell culture performance that we needed. Next, please. And we're also looking to assess the risk with the practices on our current practices related to cell bank testing and process bulk and bulk. And we also look into the in-process viral testing method. In this case, we're looking in PCR viral detection method and with a specific high-risk species in mind. And we emphasize those tests need to be turned around timely to support decision before harvest. So we take the sample at a specific time before the scheduled harvest, and we need to have the time enough to turn around the test result, and if in doubt, have sufficient time, allow for verification tests. And we also put a great deal of emphasis that we need to minimize the false signal in both positive or negative. And we also look into investigation tool if signal come in that is outside of our expectation. And when this case, we also proactively look into a mechanism within our organization, how to make decisions, define who is responsible, who is accountable, who needs to be consulted, and who needs to be informed with a RACI diagram for each step of the technical and business decision. And if you could just click on the PCR test decision tree icon. In this case, in the interest of time, if we cannot get to it in the next few seconds, that will.
Yeah, I'm sorry, Dr. Yi, it doesn't seem that the tree is showing for me.
Oh, okay, then let's just move on. And another area that we look into is the viral clearance database from our in-house experience. As Kate mentioned, it's very important that you have a good viral clearance capability built in within your purification process, not to wait for it and find out many months later that you don't have sufficient capability. So what we want to take advantage is really to clearly understand the clearance capabilities within specific purification steps and assess how the process parameter affect the viral clearance lot reduction value. Then use that to guide our purification process design, and also identify gaps and opportunity in process design phase to pursue the QBD spirit and understanding in the process. Next, please. So here's one of the examples of us looking into our virus inactivation by low pH treatment and looking into 30-some studies on XMU-LV clearance in lot reduction by low pH. And what we see is within the X side, the pH of the treatment, and we can see that when the value is greater than a threshold, we can consistently deliver desirable retrovirus lot reduction value. And once it's beyond that point, we start to see that RV value gets dropped and it's no longer in the robust range. So from process design point of view, it's important for us to communicate to the design scientists to stay in the range where the process can deliver effective viral clearance and give us a robust process performance. Next, please. And another example is to look into chromatography process conditions and how it affects virus clearance. So first, look at the left panel. We're looking at a chromatography step with a process parameter A. And looking at a wide range of value, we can see that once the process parameter is above a threshold value greater than X at the bottom of the graph where labeled as X, then we can consistently reach acceptable log reduction. So with that, again, it gives us a good understanding of where to shoot for within the process when looking at designing this particular process step. And then within that area, once we have the parameter A in the X range, we look into another process parameter, the parameter B, how that would affect the process performance in terms of virus clearance. And what we know is as long as the process parameter A is greater than X, then we have quite a consistent process performance in terms of viral clearance between the value of Y and Z units. So it gives us a pretty good design space to operate for this chromatography step. Next, please. And we're also looking to risk assessment of facility cleaning procedure. And in the interest of time, I'm not going to talk through this list, except what we want to do is really to assess what is the likelihood and impact based on the risk criteria that we had in looking at our various cleaning procedures, and that helps us to establish the priority and develop a plan for action. And once we have those necessary action in place, we'll document the relevant procedures and the studies for serve as our future reference. Next, please. And we also take into consideration of various cleaning agents and the effectiveness against lots of adventitious agents and their resistance. So as you can see here, some of the bacteria are pretty sensitive and susceptible to the cleaning agent. And then when it gets to some more resistant viruses, such as the non-lipid and small virus, such as the parvovirus and the poliovirus, that in the middle of this diagram. And then go further is to the bacterial spore that is highly resistant. So in terms of the disinfectant level, then we need to correspondingly increase those strains to have an effective control and cleaning procedure in place. Next. And so with that, back to what I mentioned earlier, in looking at the facility risk assessment, first we predefine the risk criteria and look into the relevant risks associated with each surface type, the cleaning procedure, and viral cleaning mechanism of action, and develop a preliminary risk score and ranking, and help us to establish priority and take action and document the necessary relevant procedures. Next. So I would also like to just take a moment to share my experience in really leading an integrated viral safety risk mitigation program. And we get tremendous support across the organization. Everybody, when they hear about the potential impact of what an adventitious agent contamination could do to the company and to the business, we had no problem to engage stakeholders to work with us and really to develop appropriate programs. And we get strong recognition and also get diverse functional expertise within the organization. And we also have access to external resources that help us. However, there's always challenges associated with such a program. Well, everybody recognizes it is important to have an effective procedure in place and have a mitigation plan together. Alignment on priorities are challenged, and perception and understanding on the risk different from organization to organization. And it is very difficult to calibrate low risk when the risk is extremely low. How low is low enough? And how do you take that into your planning and action? So it is a balancing act of effectiveness, the cost, and effort. Next, please. So our overall goal is to have a program in place, have the prevention and response plan be proactive rather than reactive, and we develop the necessary overall safety plan and work on the relevant SOP decision trees, material flow, and containment procedures, and also, last but not least, to have a communication plan in place, both internal and external. It's very important to have some of this plans thought out. So in summary, adventitious virus contamination is a high impact risk to any biopharmaceutical organization that produce products from mammalian cell lines. Rare event of viral contamination has caused severe impact to the companies on product supply, production facility downtime, and significant business loss. Therefore, effective risk mitigation and prevention through integrated cross-functional efforts are key.
Thank you very much, Dr. Yuling Lee, again, as well as Drs. Jerry Ann Booth and Dr. Catherine Bergman for all joining us with their expertise for today's webinar. We'll now move into the Q&A part of our program. And we don't have much time left for today's session, so we will try to address just a few questions, and we will provide a written response to all those questions that have not been answered. In any case, an email will be provided to you, which is pha@lancasterlabs.com, that you'll be able to send in any additional questions that you have, as well as if you have any a request to review the slides from today or the recording. So I will just pose a few questions that we do have time to address today. The first question would be, if manufacturing in plant cells, do we need to perform any testing for adventitious viruses? And I will unmute you right now.
This is Kate Bergman. I'll address this one. Whether you use plant-derived materials as a raw material or whether you're manufacturing your product in plants, the same issues apply. You need to do a thorough risk assessment of your production or of either of your raw materials or of your finished products. You need to think about where are these plants growing. Are they in a field or in a greenhouse? And what are the controls in that area? You need to look at what kinds of materials you're using to produce them. Are there any raw materials that you're using to produce these things that could possibly be exposed to any animal virus contamination? If you can assure that your plants are in a very clean environment, such as a greenhouse under GMP type control, and that your raw materials are non-organic and not exposed to any animal risk of animal contamination, then you probably do not need to do any adventitious virus testing. However, if you find any raw materials that do have any risk, or especially if you're growing your plants in a field, then there's animals all around, and they could be contaminating your plants with things that they leave behind, which would definitely include viruses. we do actually have experience in finding animal viruses in plant-derived materials. So it is all based on a risk assessment of your material and then an approach to addressing whatever those risks may be.
Thank you very much. And the last question is a question I received more than once, so I'll go ahead and ask it, is what do we do if we find an adventitious contaminant? What should the next steps be?
This is Jerry Ann, and I'll respond to that. First and foremost, contain the event. So, for example, if you have a suspected contamination event in your bioreactor, do not let that material go into downstream processing. And it's a good point in time for me to also remind you that during your manufacturing process, it is essential that you collect and store retains at each of the manufacturing steps, as these retains will be invaluable to you in pinpointing where the adventitious contaminant entered your production stream. Obviously, after containing the event, getting a handle on things, you're going to want to launch a full investigation, identify the contaminant, and quite honestly, this is where some of the new rapid methods, the chip methods, will come into play. You want to get as rapid an ID on that contaminant as possible, because that enables you to then predict if your validated cleaning procedures will be effective against that particular contaminant. We've gone a little bit out of order, but other activities you'll want to do is certainly to determine the lots of product that have been impacted, establish a root cause. Having all of this information will allow you to then remediate your upstream processes in order to prevent future occurrences. It will give you an opportunity to reassess your cleaning strategies and your in-process lot release testing regimen, and finally it will enable you to properly clean the facility so that you can begin manufacturing once again.
Thank you very much for everyone for participating in today's conference, and please join me in thanking our speakers, Dr. Jerry Ann Booz, who's the Director of Biopharmaceutical Services, as well as Dr. Catherine Bergman, who's the Manager of Viral Safety and Viral Clearance Services, both at Lancaster Laboratories, as well as our guest speaker today, Dr. Yuling Li, who's a fellow at the Biopharmaceutical Development at MedImmune. Again, thank you all very much for your time. We hope that you found this conference informative, and that you all have a great day.
The organizer has ended the session, and this call will be disconnected.